Good morning, everybody, and welcome to another webinar with AgroPlus. This week we have Stoller, Canada, with Andrew Bisignano. Correct me if I pronounce that wrong. And so Andrew has been working with Stoller, and he's got some exciting new products that we want to chat about today. So as far as housekeeping goes, keep yourself muted and your video off if possible, but feel free to turn both on if you have any questions that you want to ask. If you don't want your name or video shared on YouTube later, I will blur those out so no one can see who's there.
Hey, Lau.
Oh, there's Brent. Good morning, Brent. So with that, Andrew, I guess you can get started.
Yeah, so I'm Andrew Bisignano. Just to give a little bit of a background, I originated in Manitoba and learned agronomy from Cargill. Also took my my degree in agriculture business over at the University of Manitoba. Then I moved over to Saskatchewan and was trained by the Ultimate Yield Management Institute, or the RAC, doing agronomy there for about 3 years. Moved over to Alberta, worked for Farmers Edge, and then got hired on by Stoller, but we integrated with Corteva in the last July, so last year. And now we're just known as Corteva Biologicals. It's the same products as Stoller, but now we manage also their Utrisha N portfolio as well too. So let's get into a few of the products today. So we're going to kind of go through a few things, but we're going to go through, okay, who am I? What's my position? What do I do? The fundamentals of biologicals. We're going to get into 3 main products: BioForage, x-cyte, and Sugar Mover. And then we're going to get into some of the objections that we face in the marketplace, and what that all means, and what we've experienced so far, etc. We're also going to get into some trial protocols, as the start, grow, finish, and how those kind of turned out this year, and what I think is going to be important for you guys to understand going into this growing season. So my position, I'm the Biologicals Innovations Manager, and my main goal is to just be the most trusted partner that you guys have for biologicals. So if you have any questions about biologicals, if it's our products or competitors' products, just give me a call. I've got a great team of people. I've got years of background in agronomy and myself. So I'll try and get you the my understanding. But if I don't understand, I'll tell you and let you know that I'm going to go ask another person, like Mark Belmonte at the University of Manitoba, or it's my manager or my senior agronomist Rafa, etc. So I'm always going to try and find you the most trusted information is my goal. Now, the fundamentals of biologicals, we have things that are in our control and we have things that are outside of our control. What's our...
I think he just logged in.
Oh, gotcha. So we have things that are within our control and things that are outside of our control. The things that are outside of our control, of course, are photosynthesis, how much sunlight we get, and how much moisture we get. Now, things that are within our control, of course, are maintenance of our fields, or nutrition going into the fields, and then also our ability to handle stressful losses. So that could be hail events, the phytotoxicity of herbicides. It could be, a variety of different things. And these losses, if we can minimize the impact of, for example, heat blast, instead of losing 10 bushels down to 2 bushels, well, that's kind of our goal with a lot of these products, is to limit the loss of these stressful events that we haven't had solutions for in the past. And that's what we're trying to provide, is new solutions to these common problems. Now, this here is the hormone cycle. This is something that has to happen throughout a growing season in order for you to get yield. You have to have cytokinin, auxin, gibberellic acid in order to tell the plant to grow strong. You have to have ethylene and abscisic acid at the end to tell the plant to go into reproduction and senescence. Now, the way that I learned this stuff was thinking about a hockey player doing steroids. And if you think about Connor McDavid, for example, if he does steroids, injects himself with a testosterone hormone that tells him to get bigger and stronger, then he's also going to have to eat properly, drink water, and practice or work out. And then he's going to get bigger and stronger muscles, or bigger and stronger cells. And that's exactly what we're doing with these plants, is we're telling them with cytokinin, auxin, and gibberellic acid, the steroids, I would call them, we're telling the plant with those hormones to get bigger and stronger. Now, in order to support that signal, we're also going to have to provide the plant with all the nutrition, all the water, and apply these hormones at the right time in order to get the best results possible. So if you think about somebody doing steroids and what they would have to do in order to get bigger and stronger, just treat your plants the exact same way, and that's how you're going to find the best results possible. Now, to dive into each of these hormones individually, auxin is for roots, cytokinin is for branching and flowering, gibberellic acid is for overall cell sizing, and then ethylene is stress. And if we think about those four main things, and just remember those four main things, then we're doing better than 95% of the market out there.
And
A good example,
mostly like stress fighters is usually how we think of those, right?
yeah, I would even take it a step further. Think about like manipulator, for example. Like manipulator, it keeps the plant short. And what it does is it's an application that tells the plant to stop producing as much gibberellic acid. So if we tell that plant with manipulator to stop producing gibberellic acid, we keep the plant small, right? And then we're not going to get as much lodging. So there's different ways to think about this stuff, but really what we're trying to do is tell the plant to get bigger and stronger with the applications that we have, right? We give the plant gibberellic acid, we give the plant cytokinin, we give the plant auxin. And we're trying to tell that plant to get bigger and stronger when we want it to. So if we have good growing conditions like last year at the start of the season, you could tell it to get bigger and stronger at that period of time. Or when we had that heat blast, you could also tell that plant to act normally during that heat blast with a cytokinin application so that you don't lose that yield. So there's a few different ways to think about this stuff, but if we just think of it as getting rid of stress, we might be skipping out on a few good applications kind of thing. Now, if we dive into it a little bit deeper, we kind of get into that manipulator example that I was talking about. Auxin is cell initiation, cytokinin is cell replication, gibberellic acid is cell sizing. And the reason why I want to dive into that is because of that manipulator example. So we've already talked about that. So we'll skip over this slide. So when we're trying to apply these hormones and utilize this knowledge is really during three main applications. There's applications for germination. We're going to kickstart that plant to have more uniformity. We're going to have quicker germination and also overcome those cold soils. During herbicide timing, what we're trying to do is promote canopy and root growth. If we promote bigger canopy, then we have bigger solar panels, right? Conduct photosynthesis at a higher rate, catch more sunlight. We're going to have more energy to build yield. If we have bigger roots, then of course we're going to tap into more of that nutrition that we're investing into the field and pull more water. The other application that we talk about at herbicide timing is reducing herbicide flash. So if anybody's applied Viper or Odyssey, etc., that yellow flashing that happens on peas, we can just get rid of that and tell that plant to, grow normally through that.
Yep.
At fungicide and flowering, we're reducing fungicide flash and reducing the impact of heat blast. So we'll get into that in depth today. And then what we're also doing too is promoting flowering and better seed set. And that's our main goals at fungicide and flowering timing. What I also want to mention too is we're also investing a lot of research into biological protocols. So kind of a start, grow, finish, right? If we apply two applications, one at herbicide timing and one at finish, we want to research this stuff to the point where we can say you're going to get X results and we're going to warranty that. So that's where Corteva is going with this, is we want to research this to the point where it's warranted in the exact same way that herbicides are, right? If you go out and apply a herbicide and it doesn't work at all, then of course somebody comes out, they took a look at the field, see that it didn't work, and then provide you with some type of compensation. Or it would go the opposite. They would show you that it did work, right? And that's what we want to do too. We want to understand this stuff to the point where we can invest into a protocol that gives you the exact amount of yield that we deliver. So let's get into a few of these main products, those three ones that I highlighted at the beginning. So at the grow stage, or at that herbicide timing, we're really looking at one product. We're looking at BioForge Premier. And what this product is it's cytokinin, nitrogen, potash, cobalt, and moly, all packaged into one jug. And the goal of this product is to reduce stress. So I'm sure some of you have probably heard about it. When you get hail, you throw on BioForage Premier, and it's going to get that plant to wake up again after that hail event. Because very similar to when you guys go out and you cut your grass, you're cutting that grass and causing some physical stress to that grass. That fresh cut grass smell is a hormone called ethylene, that stress hormone, that's telling that plant to shut down and go into defense mode. When that plant is in defense mode, right, when some type of physical stress like hail has occurred, or that phytotoxicity from Viper or Odyssey, that yellow flashing occurs, that plant is shutting down and going into defense mode. And depending upon the amount of damage, it could actually try and push yield early. And that's what we don't want to happen. What's also happening in that defense mode is it's not really taking advantage of the growing conditions. It's shutting down. So you're not taking advantage of every growing degree day possible. So what we do with BioForage is we spray this on after a hail event or with our herbicides, and it tells that plant to grow normally through that stress, recover from that stress, and wake up again quicker. So then we're taking advantage of every growing degree day and getting the most yield possible out of this plant. So that's the goal with this product. And what I would suggest is get into that field within 24 to 72 hours after hail and spray this product on, and you'll see some fantastic results. At the finish timing, what we're going to be looking at is a heat blast now. So what heat blast is it's any time a field goes through, a canola field goes through temperatures beyond 28 degrees. And when that field is going through a 28 degree or higher temperature, it's very similar to me. I'm a bit of a ginger. If I go into a middle of the field on a 30 degree day, I don't like it very much. And it's the same thing for these canola plants. If it gets super hot, 30, 31, 32, or we even experienced last year 35, we go through a hormonal deficiency in our canola plants or our pea plants as well. Cytokinin starts to drop. And as cytokinin drops, then we get a higher rate of flower abortion. If we lose our flowers, then we also lose our pods, and we start to see a yield difference due to that heat event. So what we can do is we can apply a product called x-cyte. And x-cyte is that jug that we're going to be raffling off today. So I would suggest applying this onto your canola fields at a certain period of time, and we'll get into that today. So cytokinin, that's the only thing that's within this product. And our goal is to apply this kind of like sunscreen, right? We apply sunscreen before we go to the beach and not after. We have to apply sunscreen before, because there's no way to apply sunscreen once you're burned and then get you to get rid of that burn. So it's the same thing with these canola plants. We have to be applying this x-cyte or cytokinin before the heat event happens, and then we're going to get protection for 7 days. What I would say as well too is that let's call it 90% of our pods or our yield is created within the first 50% of our flowers. And that's something that the Canola Council of Canada released. So my thinking there, my theory is that we have to be protecting that plant in that first major heat event in order to get the best results possible. Or in the first 10 to 30% of flowers.
And
now let's get into some of the
mixing
trials.
with any fungicides or anything like that, right? Everything good on that end?
Yeah, no compatibility issues. This can just be mixed right in with your fungicide and applied in that same pass. We're going to get into a bit of like what I've seen in trials and the importance of timing today. So if like it does pay to spray it separately if the timing is necessary. So we're going to kind of get into why today, but it does mix with all fungicides if that timing works out. So these are some of the third-party trials that we went through this year. Not great in my opinion. We have some good results, some bad results, some just okay results. And I want to be honest with you guys, this is all over the place. And I was super worried back in September. I was like, okay, are we having production issues? So we looked into production. We made sure that all the product is consistent. And then we looked at, okay, is there applicator error happening? Like, are we applying this stuff at the wrong time and causing these bad results? And once we started to dive into the applications, like for example, the good application that you're seeing now on your screen, I'm going to see if I can get a pointer going, laser pointer. So each of these bars is 1 growing degree day. The orange bar is the daytime temperature, and the blue bar is the nighttime temperature. This is 28 degrees, so that temperature line where heat blast starts to occur. So this is a major heat event that we need to be protecting against. The grower applied the x-cyte the day before this major heat event, and he saw a 7 bushel difference. So perfect. That's exactly what we want to see. Just an okay, or not even an okay result. This is a bad result in my mind. Like 0.7 bushels is not what we want to see. There's no return on investment there. So when did the grower apply it? So if you look at when he applied it, the next heat event that occurred over here is about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 or 13 days away. So remember I said this only covers you for 7 days. So this application of x-cyte was kind of like throwing money to the wind, right? There was just no need for it at that period of time. If he applied it here, or maybe if it was here, maybe he would have seen a larger difference and protected himself against this heat event or this heat event here. Another good result, we see here that the grower applied it the day before a heat event, and he saw a 5 bushel difference. And we also have some trials that I want to show that we did at the canola, Corteva canola production site down around Lethbridge there, because it also reinforces this theory as well too. So we did an x-cyte and bioforage. So we did bioforage at herbicide timing and then x-cyte at fungicide timing. This orange bar is the x-cyte application. The bar over here is the bioforage. This beige one is the bioforage application. So you can see here that this grower applied the x-cyte maybe 2 or 3 days before this major heat event of 35 degrees. And he got quite a yield difference. And we'll get into this analysis here now. So we did it under pivot, and we had applied on the right-hand side and the unapplied or the check strip on the left-hand side. We omitted these areas here for spray drift, and then analyzed the right-hand side and the left-hand side and broke it up into subsections. And the reason why we want to break it up into subsections like this is so we can analyze 1 versus 1, 2 versus 2, 3 versus 3, and have more replications. Of course, yeah, more replications, you have better data. And then we dove into the actual results, and we saw that there was about an average of about 4 bushel difference. So that's exactly what we want to see, applied at the right time before a major heat event, and then we get the yield difference that we're looking for. So this was, in our minds, statistically very consistent with what we want to see. We also did another application, another pivot. This one was just x-cyte. There was no bioforage application at herbicide timing. It was applied around the exact same time, so about 3 or 4 days before a major heat event like this. Same heat event, coincidentally. The applied here, this is the unapplied check strip. And we're going to break it out very similarly. So accommodating for spray drift, we're omitting the middle, and then we're analyzing 14 versus 14, 23 versus 23, etc. And here we saw a very similar result, about a 5 bushel difference. Actually, this one, a mean difference of about 7 bushels. So this one was a very good result. So this is exactly what we want to see. So what I would take away from this is that if you have a heat event coming, and we're very good with the weathers to predict heat events, if you look on your weather application and you see next week there's going to be a heat event, I would plan for something like this. If we can get a 3, 4, 5, sometimes 6 to 1 return on investment, it's very worthwhile to look at an x-cyte application to protect yourself from that heat blast. So somewhere between 1 day before to 4 days before, that's going to be your window, and you're going to get protection for about 7 days.
That's some pretty significant results. It looks pretty good as long as we can be relatively accurate at predicting weather that far out.
Yeah, exactly. Yeah, from what I've noticed, it's really tough to predict rain. but for some reason we're very, very good at protecting or predicting temperature. Like we seem to really know when that heat blast is going to be coming. So just keep checking your weather data, and we should be able to find good application windows.
Andrew, so fair to say canola, does it work on peas as well? Any flowering crop, would that be safe to say, or flax, or which particular crops to focus on?
Yeah, we've only focused on this product within canola. We've had guys as well applied on peas, but what I would say is there's this next product we're going to talk about called Sugar Mover Premier. That is probably a bit of a better fit when it comes to peas from what I've seen. There's two main reasons. There's one reason because it has cytokinin in there, so you are going to get heat blast protection with this Sugar Mover Premier on peas. And then it also has boron in there. And boron is super important for peas when at flowering timing. The reason why is because it's the movement of sugars into reproductive material that we're trying to focus on here. And peas, for some reason, when they get super stressed, like peas would be more comfortable dead than they would be alive, in all honesty. It's almost like they want to abort some of the peas in that pod. So what we're doing with this Sugar Mover product, with the boron and the cytokinin, we're protecting against heat blast, and we're moving sugars into those pods. So then we're getting a higher average of peas in the pod. So instead of seeing those 4s and 5s, what we've been seeing is more like 6, 7, 8s, and 9s. So we're just trying to increase that average and maintain that pod set, get rid of the stress of heat blast, and move those sugars into focusing on the reproductive material instead of the building a bigger plant kind of thing. Does that answer your question?
Yes, thank you.
Yep, no problem. Yeah, so that's what I would suggest if we're going to focus on peas, especially because I've started to hear that we're using less herbicides on peas and lentils down in the south there, and going with more of the residual products. I would probably recommend focusing on Sugar Mover at that first fungicide timing and throwing this in. That would probably be the best application for peas now. If you do still have guys that are going out with Viper Odyssey, then I would focus on that BioForge product.
Probably one of the hurdles will be guys wanting to go in if it's just this product. So throwing it out there just for discussion, perhaps. Yeah. Is it a legal product to spray by drone?
Yes, these are legal products to spray by drone. I just don't know how good of an application those are yet. Like we can do some trials and figure that out together, but I would just caution that I haven't seen the results because I haven't played around with it yet. It's not to say that it won't work. I just don't have a solid answer for you if it will work or not.
Okay.
Yeah. So these are kind of the main products. We didn't go through all of them today. We went through BioForge, Sugar Mover, and x-cyte. We also have Nutritia, and we also have Stimulate. So if you guys have any questions about those, just give me a ring and we can go through those more in depth. But I also want to talk today about the objections that we face in biologicals. Because I think it's safe to say that there's an elephant in the room when it comes to biologicals. And those main 3 elephants are, it's snake oil, I haven't seen the results, and these are being sold like silver bullet products. So let's get into these individually, because I think once we start to dive into them individually, we can start to understand where a lot of growers, where a lot of retails, etc., people are coming from when they're talking about this stuff this way. So the it's snake oil objection, I completely understand where guys are coming from. This is a market that is bloated at this point. There's nutrition products, organic acids, amino acids, there's plant growth regulators, microbial agents, seaweed extracts. There's so many different things classified as biologicals that we now have about 1,200 different companies in this landscape talking about this stuff and trying to sell in this division. And I can completely understand where growers and retails are coming from when they start to call it snake oil, because sometimes people are going into this stuff and trying something that, might be made in a bathtub, might have no certifications, right? Or something that really just shouldn't be used at that period of time. So what I would say is, in order to kind of talk about why people are, or how to get somebody over calling it snake oil, would be just to ask the right questions, right? It's what were you trying to use that product for? What was your overall goal? Did we apply it correctly? Where was it sourced from? Was there any sort of good data backing up this application? And we can start to really dive into how it was used, and how it really isn't a one size fits all approach, right? There's going to be a lot of different things factoring into these applications that we need to, even with that x-cyte application that I was talking about today, we got to factor in when it's applied, what the temperature is, how much moisture we have. If we're in drought conditions, we shouldn't be using this stuff. There's just a lot of things to be factoring in. And I can understand when a grower comes to me and says that, hey, this stuff is snake oil. And usually we can have a pretty upfront conversation about it. The main ones that we don't play in, or the biological categories that we don't play in, are nutrition, really, amino acids, fulvic acids, and humic acids. We do have a bit of nutrition mixed in with our hormones, but these ones are kind of like the outskirts of the market that we don't play in. So fulvic acids are going to be kind of like carriers. They loosen up the cell wall to get things into that cell wall, like nutrition, hormones, etc. It gets things to where it needs to go. Amino acids, they're kind of like the building blocks of proteins, or they exactly are that. They're building blocks of proteins. So I can kind of understand where some businesses are coming from, that we should be adding in some of these things. I just haven't seen those results. So I would just ask for as much data as you can get from them. Humic acids, they're a carbon-based product that goes and mixes into the soil, and in hope to release some more of the nutrition that's being bound to different things. What I would say is that these things, like you heard me say today, are very different, right? They're all, though, amino acid, fulvic acid, and humic acid. Now, they're applied in very different ways. So just having that basic understanding of what amino acids are, what fulvic acids are, and humic acids are, you can probably ask a grower, okay, did you spray your humic acid onto your plants, or was it mixed into the soil? And if you sprayed it on, then you probably wasn't going to get any sort of result from it. So just having that basic understanding to ask the right questions and tell a grower yes or no is probably going to be the best way to get him over that snake oil hump. These are the main ones that we play in. We don't have seaweed extracts, but seaweed extracts, their main goal is to extract cytokinin from that seaweed to be applied in a jug worth of product. So cytokinin we talked about today, it does work. It works in different scenarios. What I would say is that they don't have a guaranteed analysis due to this extraction method. So that's the only concern I have with it. They do work, is what I would say. It's just we don't know when they are going to work, because we don't know how much seaweed extract or cytokinin is within that product. The microbial agents and bacteria agents, that's kind of like Utrisha, Invita, etc. These are or inoculants, right? These are biologicals that help the plant to create something, if it's nitrogen, phosphorus, etc. Plant growth regulators, this is where we play. Plant growth regulation can go in up regulation or down regulation. We can tell the plant to grow bigger and stronger, or like manipulator, we can tell it to grow smaller. That's where plant growth regulators are. The second objection, I haven't seen results. In my opinion, we need to start teaching growers what to look for, because in all honesty, like these pictures here are pictures that I took. We went to our small plot research in Strathmore, and visually, I couldn't see the difference from one plot to the next plot. But when we started to dig up these plants, wash off the root systems, and show off the physiological changes, like more branches, bigger root system, more pods, etc., that's when I could see the difference. So what I'm trying to say is that growers, we can't be scouting our fields at 40, 50 kilometers an hour and driving by. We have to go into that field, pull out some plants, and see the difference. And that's where we're going to really be able to see the difference that biologicals are causing. So these are some of the pictures that I took. You can see here the plants that we dug up. We pulled off some of the leaves just to show it off a little bit better. There's 12 branches over here, and then there's 8 branches over here. A bigger root system as well too, and more pods. And remember, I couldn't see the visual difference from one plot to the next plot. So what I'm trying to say, super important, pull some plants out and look at the physiological changes that are being caused and not just, from the side of the road. What I would say as well too is if you've ever soil sampled before, we always talk about doing a good composite sample, taking samples from the average points in the field. So when you are going check versus treated, like if this is the treated and this is the check, always try and pull from those average points and do a direct side-by-side comparison. And adding some type of whiteboard with a graph, that's going to be the best way to show off those results or see the results. The other one is the silver bullet product. So I can completely understand where growers and retailers are coming from. There's a lot of over-promising happening within this marketplace. I'm sure everybody went to either AgriTrade or went to the Lethbridge farm show last year and walked around and saw all the different biological companies. And everybody seems to be promising, oh, you're going to get 5 bushels from this application. And I had a couple of growers that I was walking around with, and everybody was joking that we should go buy 25 of these products, and each of those are going to give me 5 bushels. Well, then I'll just be making infinite yield, right? Now, this isn't the way that we should really be selling this stuff, right? Like we can't be over promising. In all honesty, I can promise physiological change. I can't always promise a guaranteed yield. We want to get there at some point, but from what you saw with my even results with x-cyte today, even the good results, they were a range from 4 to 7 bushels. I can't really tell you why, like maybe it was nutrition, maybe it was some other factors. But the thing is that we're going to cause the physiological change to give you a higher probability of success. If you have bigger branches and a bigger root system, you have a much higher probability of success than you would if you didn't have those things. So our goal is to, of course, get to the end where we can say this protocol, it's going to give you a guaranteed amount of bushels. But right now, physiological change is what I can guarantee. And then those 4 to 7 bushel range, if we apply it right, have good nutrition, have good water, right? Lots of factors we got to take into consideration in order to get that end result. Do we have any questions before I move on?
I would say, like myself doing some small plot trials in canola and stuff last year with different growth regulators and growth promoters. Yeah, absolutely. Like what you were saying about the physiological changes, it can be quite dramatic with pod counts from one plant to another. If you pull five plants out of a plot versus another 5 plants, it's amazing some of the differences.
Yeah, completely agree. Yeah.
When I add there, Andrew, maybe you can comment more on it, but it's not always about yield. Like we've seen results where, like, correct me if I'm wrong, we've seen like better plant stats, or like not plant stat, standability within the crops, right? So that helps ease harvest at times. So we're looking at that too, not just yield. How do we make your guys' lives easier with these products?
Yeah, exactly. Yeah, so true. Like the nice part about that sugar mover is it also helps with standability, right? If we can tell that plant to with cytokinin to build more cells, like do cell replication and get a stronger stem, then of course we're going to have less plants that fall over, and will make your life a lot easier when it comes to harvesting that plant. So there's various things that we're looking at, like this stimulate at the start, for example. If we have more uniformity caused by this stimulate at germination timing, well, then maybe we have an easier harvest. Or another way is maybe this stimulate, it helps with germination and getting that plant out of the ground quicker and grows through those early stages quicker. If we think about canola in that cotyledon stage, well, flea beetles are attracted to canola in that cotyledon stage. What if we got that canola plant to grow through that cotyledon stage a lot quicker by having stimulate there? Well, then there's probably going to be a far less need for a lot of insecticides to be sprayed. So there's different things to take into consideration when we're applying this stuff, but yeah, the ones that we talked about today, heat blast, movement of sugars into peas, bioforage, let's get rid of stress at, that herbicide timing or hail. That's kind of my main fits. I do mention stimulate a couple times here, but we just didn't talk about it today, so let's not get too in depth on those ones. I always mention some foliar fertility, and the reason why I mention that is, if you took agronomy or remember the days of learning agronomy, there's that picture of a barrel, holding water, and all those slats on the side, they represent all the nutrition that a plant needs in order to grow. And if one of those slats or one of those pieces of nutrition is low and we have a limiting factor, then the water pours out and we don't get the yield we're looking for. So adding in some foliar fertility during these applications to get rid of those limiting factors is always going to be a good agronomic approach. Even though we don't have anything on the market with this stuff right now, I would say it's always a good idea to be looking at some good foliar fertility. So those pictures, I just wanted to mention those pictures that I showed earlier. It was stimulate at herbicide and then x-cyte at first fungicide timing. That was the protocol that we went with, those pictures that I showed. These ones here. And then the peas, my recommendation would probably be bioforage and then first fungicide timing. I do say x-cyte, but sugar mover, from what I've seen this year, was a better fit than the x-cyte. So I should change that, excuse me. But this was what we did at our Strathmore site, was that bioforage plus x-cyte and some foliar fertility. And these were the results that we saw. More branches, more opportunity for pods, etc. I also have this as a bit of a cheat sheet. So this tells us a lot about when to not use this product or these products, what crops to use these products in, and when to use them. One thing I do want to note is if you're in dryland drought, these products are probably not a great fit. I would stay away from using them during a drought time. But let's say that we have pivots or we've had moisture in the past that the plant is still growing on, then we can apply this stuff. So just wanted to make that abundantly clear. But yeah, thanks for your time today. I hope everything was as clear as possible. If you have any questions, my contact info will probably get sent out. And don't hesitate to give me a call if you do have any questions. Anything else anybody would like to ask or add in?
I would say your last comment about drought was very good. I saw that a little bit in some of my plots in wheat last year as well. Some of those early season hormonal products where we encouraged more root development and stuff to start with ended up hurting us in the end when things dried out. And it wasn't a great result for some of those products. Yeah, we do see that every once in a while. If the products are applied at the wrong time during drought, we can actually cause a bit of a lag in yield. So something to take into consideration is make sure we're doing exactly that, trying to determine how much moisture that plant has to live off of.
I'm going to ask the group, setting aside rain, because we can't solve that. What other environmental challenges have you guys faced, or environment things that affected your crop, like where you have that lodging, stuff like that, for example? Does anyone have anything?
Well, there's always some lodging out there, and it seems like every year at least one or two of our customers gets hit pretty good with a hailstorm. So there is definitely some potential there.
Mm-hmm.
I would also like to add for anybody who's listening in, I have great faith in these products. It is really nice to see a company like Corteva and Stoller together, working to actually have some accountability in this field with some published numbers. Like your one slide you mentioned, when it comes to seaweed, not all the sources are the same. Lots of these products, they're guaranteeing, we got so much seaweed in there. They're not really telling you what the components are of that seaweed. And even if it is the same seaweed, if it's harvested in the summer versus the winter, it's going to have a different content in it. So I'm really glad to see you guys doing this, and hopefully it's going to cause a little bit more kind of accountability in the marketplace for these products.
Yeah. Yeah, that's definitely the goal, is to get some accountability over the next, 3 to 5 years that everybody has to respond to, right?
Absolutely. And I'll be putting your products in my small plot trials this year, so it'll be interesting to see how those fare up against some of the competition.
Awesome. Awesome.
Anybody else have anything to contribute or any questions? All right, well, I guess we can go ahead and do our draw then.
A couple months kind of thing, or at least before the season.
Yep. Sounds good. We will make a note and put it aside for him so he can pick it up when he wants. Yeah.
Cool.
All right. Well, everybody, thanks for attending and watching. And thank you, Andrew, for presenting, as well as Brent for being here, although he's already left. So I will now stop the recording, and yeah, we'll get this posted and put up on YouTube here later on.
Right on. Thanks, guys. Have a great day, everybody.
Thank you.